This presentation is about the genetics of hemophilia A and B. Both hemophilia A and B follow the X-linked recessive pattern of inheritance. Genetic counseling is now offered in many hemophilia treatment centers across the country. The purpose of genetic counseling is to explain how hemophilia is passed down in a family or inherited, draw family histories to look for women and girls who might be carriers of the hemophilia gene or men and boys who are mildly affected, arrange genetic testing and explain the results, discuss family planning options. Hemophilia is just one example of a genetic disease that can be passed down in a family. Part 1 will cover chromosomes, genes, and DNA. Hemophilia A and B are caused by a mutation or change in DNA. Most cells in the body contain chromosomes that are made up of tightly wound DNA. It is the DNA that holds the instructions for how the human body works. New babies get 23 chromosomes, half of their genetic instructions, from mom, and 23 chromosomes, half of their genetic instructions, from dad. The baby ends up with their own unique set of 46 chromosomes. This is why babies have some features like mom and some features like dad. Chromosomes. Most people have 23 pairs or 46 total chromosomes in every cell of their body. The first 22 pairs of chromosomes carry the genetic instructions that are the same in men and women. Instructions on the 23rd pair determine the sex of a person. Women have 2 X chromosomes. Men have 1 X and 1 Y chromosome. When DNA unwinds from the chromosome, it is a long strand of genetic instructions that sort of looks like a twisted ladder. DNA holds many thousands of smaller units called genes. Genes are the instructions for the human body. Genes make proteins. Proteins have different jobs in the body to help keep people healthy. Factor VIII and Factor IX proteins are needed to stop the bleeding after an injury or surgery. The genes for hemophilia A and B are on the bottom of the X chromosome. F8 for Factor VIII is the gene name for hemophilia A. F9 for Factor IX is the gene name for hemophilia B. The X chromosome has about 1,400 genes and is 3 times larger than the Y chromosome. The Y chromosome has about 55 genes and no genes for hemophilia. Mutations, or changes, or variants, these terms mean the same thing, in the DNA of the F8 and F9 genes decrease the ability of the blood to form a clot. This is why people with hemophilia bleed easily. The DNA alphabet has only 4 letters, A, T, C, G, that are read in groups of 3. Genes are like words and sentences in a book. If the letters or instructions are present and read in the right order, the right protein will be made. Instead of using the DNA ATGC, we will use the English alphabet to make words we can follow. If the DNA is meant to spell the word red, but the E is changed to a D, the letters RDD may cause the gene to not work properly. If the sentence is meant to read, "The car was red," and car is missing, then the sentence, "the was red," may cause the gene to not work properly. These are two common examples. However, there are many types of DNA mutations. Genetic testing will give the exact mutation or change for each person. Knowing your mutation may affect treatment decisions and participation in research projects, allows for family planning, prenatal screening, and testing decision, allows women and girls in the family to get carrier testing. Part 2, Inheritance, or how a genetic disease is passed down in a family. Hemophilia A and B follow the X-linked recessive pattern of inheritance. X-linked means a specific gene is on the X chromosome. Recessive means 2 copies of the non-working gene are needed to cause the genetic disease. However, men and boys will show X-linked recessive hemophilia. If their X chromosome has the non-working gene, because their Y chromosome has no genes for hemophilia, so they have no way to make factor. Women and girls are called carriers if they have one X with the working gene that makes factor, and their other X has the non-working gene that makes no factor for hemophilia. Please remember, 1/3 of people with the hemophilia A gene have new mutations. This means the mutation did not come from mom or dad. However, the person with the new mutation can now pass it down to their children. Also, every person in the world has the potential to carry genes that code for genetic diseases. How X-linked recessive works when dad has hemophilia and mom is not a carrier. For the rest of the slides in this presentation, the blue X chromosome is used to show the working gene. The orange X chromosome is used to show the non-working gene. The Y chromosome has nothing to do with hemophilia. If dad has hemophilia, he has one orange X with the non-working gene for F8 or F9, and his Y chromosome has nothing to do with hemophilia. If mom is not a carrier, she has 2 blue X chromosomes, and each one has a working gene that makes factor 8 or 9. There are 4 possible combinations for how the X's and Y's will make a baby. Option 1. To make a boy, dad gives his Y chromosome that has nothing to do with hemophilia, and mom gives her first blue X with the working gene. This will be a son, and he will not have hemophilia. Option 2. To make a boy, dad gives his Y chromosome, and mom gives her second blue X with the working gene. This will be a son, and he will not have hemophilia. Option 3. To make a girl, dad has to give his orange X with the non-working gene, and mom gives her first blue X with the working gene. This will be a daughter who is a carrier of the non-working hemophilia gene. Option 4. To make a girl, dad has to give his orange X with the non-working gene, and mom gives her second blue X with the working gene. This will be a daughter who is a carrier of the non-working hemophilia gene. In this case, if dad has hemophilia and mom is not a carrier, 0% of the sons will have hemophilia, and 100% of the daughters will be carriers. A dad cannot pass hemophilia to a son because the gene is not on the Y chromosome. Now, how X-linked recessive works when mom is a carrier of one non-working hemophilia gene and dad does not have hemophilia. Again, blue X chromosome shows the working gene, orange X chromosome shows the non-working gene. The Y chromosome has nothing to do with hemophilia. In this case, Dad does not have hemophilia, so he has one blue X with the working gene and a Y chromosome. Mom is a carrier, so she has one blue X with the working gene and one orange X with a non-working gene. There are 4 possible outcomes for how the X's and Y's will make a baby. Option 1. To make a boy, Dad gives his Y chromosome, and Mom gives her blue X with the working gene. This will be a son who does not have hemophilia. Option 2. To make a boy, Dad gives his Y chromosome, and this time, Mom gives her orange X with the non-working gene. This will be a son who has hemophilia. Option 3. To make a girl, Dad gives his blue X with the working gene, and Mom gives her blue X with the working gene. This will be a daughter who is not a carrier. Option 4. To make a girl, Dad gives his blue X with the working gene, and this time, Mom gives her orange X with the non-working gene. This will be a daughter who's a carrier like Mom. When Mom is a carrier, her sons will have a 50% chance of getting the hemophilia, and her daughters will have a 50% chance of being a carrier. Take home points. If Dad has hemophilia and Mom is not a carrier, none of his sons will inherit hemophilia. 100% of his daughters will be carriers. If Mom is a carrier and Dad does not have hemophilia, each son has a 50% chance for inheriting hemophilia. Each daughter has a 50% chance of being a carrier like Mom. Part 3. Women and girls can also have hemophilia. Many times the question comes up about how females can have hemophilia. You may meet some women and girls with hemophilia through clinics, camps, and other activities. There are 3 main ways that a girl can have hemophilia. The first way is if Dad has hemophilia and Mom is a carrier. Dad has hemophilia, so he has one orange X with the non-working hemophilia gene and a Y chromosome that has nothing to do with hemophilia. Mom is a carrier, so she has one blue X with the working gene and one orange X with the non-working gene. There are 4 possible combinations for how the X's and Y's will make a baby. Option 1. To make a boy, Dad gives his Y chromosome, and Mom gives her blue X with the working gene. This will be a son who does not have hemophilia. Option 2. To make a boy, Dad gives his Y chromosome, and Mom gives her orange X with the non-working gene. This will be a son who has hemophilia. Option 3. To make a girl, Dad gives his orange X with the non-working gene, and Mom gives her blue X with the working gene. This will be a daughter who is a carrier like Mom. Option 4. To make a girl, Dad gives his orange X with the non-working gene, and Mom gives her orange X with the non-working gene. This will be a daughter, and she will have hemophilia. When Dad has hemophilia and Mom is a carrier, there are 4 options for a baby. A son who is not affected, a son with hemophilia, a daughter who is a carrier, and a daughter with hemophilia. Each of these 4 options have a 25% chance of happening in each pregnancy. The second way a girl can have hemophilia can occur in girls who have Turner syndrome. Women and girls with Turner syndrome are born with 45 chromosomes and only one X. If their X chromosome has the non-working gene, they will be diagnosed with hemophilia. A third way a female can have hemophilia is by something called skewed X inactivation. Women and girls have two X chromosomes, and they do not need two copies of all 1,400 genes on their X. X inactivation means that one X is turned off in every cell in girls. This is a normal process that only occurs in girls very early in the embryo. Because boys only have one X chromosome, it cannot be inactivated or turned off. This diagram shows a body filled with lots of cells for skin, brain, blood, etc. In this example, Dad has hemophilia and Mom is not a carrier. The orange cells show Dad's X with the non-working gene is active. Mom's blue X is turned off in these cells. The blue cells show Mom's X with the working gene is active, and Dad's orange X is turned off in these cells. Random versus skewed X inactivation in carrier women and girls. In most women and girls, X inactivation is random, or about equal whether Dad's X or Mom's X is turned off. However, in some women and girls, this process is skewed, or occurs more frequently in one direction or the other. If most cells have Mom's blue X with the working gene active, and very few cells have Dad's orange X with the non-working gene active, this girl will have no bleeding problems. However, if most cells have Dad's orange X with the non-working hemophilia gene active, and very few cells have Mom's blue X with the working gene active, this girl will have hemophilia. Women and girls with hemophilia have the same risks for bleeding problems and inhibitors as men and boys. They are given the same medical treatment as men and boys with hemophilia. Factor levels and genetic testing are recommended for women and girls to determine carrier status. Why both? Factor VIII and Factor IX activity can be unreliable in carriers. About 30% of carriers have low Factor VIII or IX activity, as expected. However, 70% of carriers will have normal Factor VIII or IX activity. Genetic testing will find the exact DNA mutation in a carrier regardless of the factor level. When to offer testing to potential carriers? Girls with bleeding or bruising problems, prior to surgical procedures, regardless of age, around the time of their first period. Women with bleeding problems, for family planning and prenatal screening, for knowledge of exact mutation for family. Basically, whenever a potential carrier requests testing. Hope for the future. Some gene therapies and gene editing, CRISPR, treatments have the potential for being a cure for hemophilia A and B. Research and clinical trials are currently underway. Part 4. Family planning, for parents and adults considering children. Family planning. One of the most important points to remember is that each pregnancy is independent. If the risk for a son with hemophilia is 50%, and the first son has hemophilia, this will not decrease the risk of 50% for the second son to have hemophilia. Some families have had 5 sons and all 5 had hemophilia. Decisions about having children and prenatal testing are yours alone. Couples have chosen each of the following options depending on their unique circumstances, including having no children, adoption, natural conception with no prenatal screening or testing and accepting the outcome, natural conception with prenatal screening or testing and using the testing information to prepare for the outcome. Natural conception with prenatal screening and testing, NIPS, non-invasive prenatal screening, occurs around 10 weeks into the pregnancy. This can be done by drawing blood from the mother's arm. NIPS shows sex only. It does not test for hemophilia. CVS, chorionic villus sampling, occurs 11 to 14 weeks into the pregnancy. This takes a tiny piece of the placenta for testing. This shows the sex and whether the hemophilia mutation is present. Amniocentesis occurs 15 to 20 weeks into the pregnancy. This takes a small amount of amniotic fluid from around the baby. This shows the sex and whether the hemophilia mutation is present. In vitro fertilization, or IVF, means that sperm and eggs are combined in a lab to make embryos. In the case of hemophilia, IVF can use donor eggs if mom is a carrier, donor sperm if dad has hemophilia, donor embryos where neither donor has the hemophilia gene. These embryos are not at risk for developing hemophilia. IVF with pre-implantation genetic testing. It's offered to families where the hemophilia mutation is known. This uses mom's eggs and dad's sperm to make embryos. Then, they test each embryo for the known genetic mutation. This allows couples to choose either the boy or girl embryos that test negative for the mutation. Here are resources for families. Hemophilia and the Fencer, Luke's story. This is good for a new diagnosis as well as those who are preparing to leave the nest. Hemophilia of Georgia offers a wealth of information and excellent graphics. Genetics Home Reference offers very good resources for family members who are asking for more information. The National Organization for Rare Disorders, or NORD, offers 2 pages on hemophilia A and hemophilia B. The following people created this presentation: